Greetings, students. My name is Malcolm Burns. By this point, you've already seen me for a class or two. But my goal this morning here out on my deck in suburban Maryland is to record a lecture for a flipped class. And the title of the lecture is Protein Folding, Unfolding, and Misfolding, Prions and Prion Diseases. This flipped class is a new one in the previous 2 years, 2020 and 2021. I recorded a lecture for a flipped class on the SARS-CoV-2 spike protein. But since the pandemic is hopefully beginning to wind down now, that topic is no longer as relevant. Now we have a different variant of the SARS-CoV-2 virus circulating. Latest is that it's a subvariant of Omicron called BA.5 that's gaining prominence. Omicron is less virulent, more easily transmissible. Many people in the United States are now vaccinated and boosted. So this situation we find ourselves in now in July 2022 is actually quite different from how it was a year ago and 2 years ago. You know, a year ago when I was sitting out here in late June recording a lecture, the Delta variant was just starting to surge, and now that's long gone, and there's no more Delta around. We just have Omicron. So I'm going to go ahead and share my screen now. Let me try to do that now. Hopefully you can see that screen. And so again, the title of this lecture is Protein Folding, Unfolding, and Misfolding, Prions and Prion Diseases. And you should view this lecture before coming to the flipped class. There's also a quiz, an ungraded quiz that you can take to prepare yourself for the class itself. And there's also sort of an outline of how that class is going to go. So the reading assignment is chapter 4 of Nelson and Cox's Lehninger Principles of Biochemistry, 8th edition. That's the textbook for the class. And the objectives are listed at the end of this lecture. So what I'd like to talk about is first, protein folding and denaturation. Then Christian Anfinson's experiment in the 1950s, using artificial intelligence or AI to predict protein structure, intrinsically disordered proteins and protein regions, cellular proteostasis and the protein folding process, chaperones, diseases of protein misfolding, so-called amyloidoses, prions and prion or prion-like diseases, and then pose a question: is Alzheimer's a prion disease? So let's start with a discussion of protein folding and denaturation. Shown here in this diagram is bovine ribonuclease. It's a small protein, about 124 or so. Let me check. 124 amino acids in size. And it has 4 disulfide bonds. It folds into a native tertiary structure. It's a monomer. And it catalyzes the hydrolysis of RNA. It's an RNase, so it degrades RNA. And you can place this protein in a denaturant, a chemical denaturant, and it will unfold, lose its structure. And you can also then remove that denaturant, and actually for this protein in particular, it will refold back. Now notice in this diagram here for the denaturation, the disulfide bonds remain intact. Denaturing a protein doesn't remove disulfide bonds or break them. To do that, you need a reducing agent, such as beta-mercaptoethanol or dithiothriotol. Those are reducing agents that will reduce the disulfide linkages between those cysteines, giving free thiol groups on the cysteine residues, and so that the protein can be completely unfolded. So when proteins adopt their native secondary, tertiary, and quaternary structure, they're said to be folded. Now, of course, some proteins, particularly monomers, don't fold into a quaternary structure. They just have a tertiary because they're monomeric. The folded state of a protein is in equilibrium with its unfolded state, although the equilibrium is skewed slightly toward the folded state, and that's generally true, and that's shown here for ribonuclease, where actually the equilibrium lies slightly to the left toward the folded state, and the delta G for unfolding is a small positive value, indicating that actually unfolding is slightly disfavored, whereas folding, or the existence of the folded state, is somewhat favored or slightly favored. The different kinds of denaturants. You can have chemical denaturants, such as 8 molar urea, 6 molar guanidine hydrochloride, or sodium dodecyl sulfate, SDS. These expose hydrophobic side chains and proteins to the solvent, allowing the proteins to aggregate, unfold, and then aggregate. In addition to chemical denaturants, you can have heat, you can have extremes of pH as well. Now, you can have even small changes in pH that will cause an enzyme, for example, which is a protein, to lose its catalytic activity. And there, what you're doing is you're changing the ionization state of amino acid side chains or other groups important for catalysis. But extremes of pH will cause the protein to actually unfold or denature. Now, although some proteins can fold spontaneously, others need help from foldases called chaperones. These proteins bind to unfolded or partially folded proteins. They prevent premature aggregation and thereby guide the folding process. An example of a chaperone is heat shock protein 70. There are also chaperonins, which are types of chaperones that exist as complexes of and form a large barrel structure into which the unfolded protein goes, and that large complex, whose activity is ATP dependent, will help it fold. I'm going to talk more about chaperones and heat shock protein 70 in particular in just a minute. But first, I want to describe Christian Anfinson's experiment in the 1950s, for which he received a Nobel Prize in chemistry. So what Christian Anfinson wanted to know was, does the amino acid sequence of a protein determine the three-dimensional structure of the protein? So, and the answer, just by the way, is sometimes. Often, though, chaperones are needed to help a protein fold. Right? So even though the structure may be determined by the amino acid sequence, the protein cannot necessarily fold into that three-dimensional structure without help. We'll talk about chaperones. So let's talk about his experiment in the 1950s. His plan was to disrupt the three-dimensional shape of a protein by reversible denaturation, and then ask what is needed to restore the shape and restore the biological activity of the protein. He chose the small protein bovine ribonuclease, which we just talked about, has, and here's in this box is a depiction of its amino acid sequence. It has 124 amino acids and 4 disulfide bonds. And it catalyzes hydrolysis of phosphodiester linkages of RNA. So it's an RNase. What he did was he put the RNase in a denaturant solution and included a reducing agent. And that's shown here in step 1. So here we have the native ribonuclease existing in its folded tertiary structure. And then he placed it in buffer in a dialysis bag, and then put that in a solution containing, in a big bucket containing 8 molar urea and beta mercaptoethanol. I'm going to move over a bit to get out of the sun, folks. So excuse me for a minute while I get out of the sun. There we go. So you can see that now the protein is not only unfolded through the action of the urea, the denaturant, but also the disulfide linkages are all broken by the reducing agent. Then what he did was he dialyzed the unfolded RNase in buffer containing no denaturant under oxidizing conditions, but included just a small amount of beta mercaptoethanol. Now, why did he do that? And that's shown here. It's shown here, right? So what happened, by the way, was that the protein folded, but if he hadn't included that trace of beta mercaptoethanol, then perhaps as the protein folded, aberrant disulfide bonds might have formed. Okay, so we can see that here. We can see the 2 yellow ones. Yeah, those are correct. The 2 blues coming together is correct, but we've got these aberrant formation of these other two, right? So what the beta mercaptoethanol will do will allow the protein to sift and sort among the different disulfide linkages until it forms the correct ones. And that's shown here down at the bottom, right? So that was why you include that trace of beta mercaptoethanol, to allow the correct disulfide bonds to form. And then the result. So what was the result? The result of this experiment was that although the RNase lost all of its activity upon unfolding, he tested it when it was unfolded, it was not active. It regained its activity, its catalytic activity upon folding. So the protein refolded to its active structure, which proved that the amino acid sequence alone can determine the 3-dimensional structure of a protein. Famous experiment. So now I'm going to turn my attention to something that's brand new. Hold on a minute. And that is the use of artificial intelligence to predict protein structure. And I apologize for the somewhat busyness of this slide. There's a lot of information on it. And by the way, shown on the right here, on the right, are the actual and the predicted structures of 2 proteins or protein domains. The experimental or actual structure of the RNA polymerase domain of RNA polymerase is shown in green, and the predicted one is in blue, predicted by this AI method. And then we have the tip of the adhesin protein, actual shown in green, predicted shown in blue. Amazingly accurate predictions of structure. So what happened was that in July of 2021, the London-based firm called DeepMind, which is part of Alphabet, Google's parent company, made public an artificial intelligence-based tool called AlphaFold 2 that could use a protein's genetic sequence to accurately, or the sequence of its gene, to accurately predict its 3-dimensional structure. This fulfilled a long-sought goal, which was to determine a protein structure from its amino acid sequence alone. This has been had been a goal ever since Anfinsen did his experiments, right? So what's needed to predict the structure of a protein? Now, in December 2020, a little bit of a backstory. In December 2020, AlphaFold caused a sensation by winning a competition that was held every 2 years called CASP, Critical Assessment of Protein Structure, whose goal was to solve protein structures computationally. But DeepMind did not release its software or its predictions into the public domain. So they predicted these structures, but didn't give anybody any information. So people were quite agitated, right? They wanted to be able to do this too. So what happened was that a protein biochemist by the name of David Baker and colleagues, who I believe are at the University of Washington, stepped into the breach and used information from a public talk on AlphaFold to design their own model, which they called Rose TTA Fold, that was also very good at predicting protein structures. This prompted DeepMind to release its computer code, and both models were then published on the 15th of July, 2021. So for now, what about AlphaFold 2? So far, AlphaFold 2 has predicted the structures of nearly every protein in the human genome and the entire proteomes of some organisms. Now remember, proteome is the complete catalog of all the proteins in a cell, tissue, organism, even like a multicellular, a single-celled organism like E. coli. So they've predicted the structures of 365,000 proteins in all. What's next up for DeepMind? To release more than 100 million structure predictions, those for nearly half of all known proteins. So they're really going at it now in predicting structures, and many of these are very good. But there's some limitations of AlphaFold. First, it's not good at predicting the structures of protein complexes. And so far, Rose TTA Fold is better. It was able to predict the complexes of some dimers, two proteins coming together. AlphaFold can't predict structures of proteins that can assume different conformations. For example, intrinsically disordered proteins, which make up 30% of known proteins. And then finally, AlphaFold cannot predict how mutations will affect structures. So it can't come up with a new structure of a protein that has a mutation. It can't figure out how a mutation will cause a change in structure, or might. Now, these advances using these computational approaches are only possible because of the laborious work that was done over the decades by biochemists using X-ray crystallography, NMR, and cryo-EM, because both AlphaFold and Rose TTA Fold use AI to identify folding patterns in solved protein structures in large databases such as the protein database and others. They use these existing patterns along with physical and biological constraints to predict the best structure for a protein. So they really depend upon this work that has gone before. And we have here a couple of sources of information for this. Information on the slide, Callaway, and then a service in Nature and Science magazines. All right, again, this is a rather busy slide here. I want to talk about what are known as intrinsically disordered proteins and protein regions. So it wasn't until the mid-1990s that the functional significance of intrinsically disordered proteins was recognized. IDPs, as they're called, fail to fold into stable, defined structures as free monomers, but they're capable of becoming ordered and into a particular structure upon binding to partner proteins. Now, in the absence of the binding partner, they're dynamically disordered and they fluctuate through a range of conformations. But when they interact with the partner, they then fold into a defined structure. They're characterized by biased amino acid composition. They have few hydrophobic amino acids and a higher than usual number of charged or hydrophilic ones. You know, the hydrophobic amino acids can come together and form a hydrophobic core and help that domain fold or protein fold. So there aren't too many of these in these intrinsically disordered proteins. These proteins can act as hubs in protein interaction networks. They integrate multiple signaling pathways. They act as scaffolds that bind proteins from different pathways and are important in the regulation of transcription, translation, and the cell cycle. They also play critical roles in the assembly of macromolecular machines, such as the ribosome, and they're important in chromatin organization and the assembly and disassembly of microfilaments and microtubules in the cell. Moreover, by aggregating together, they promote phase separation in the cytosol and form membraneless organelles that serve to compartmentalize cell components. Now, intrinsically disordered regions in proteins are sites for post-translational modification, for example, phosphorylation. It's thought that the protein kinases have easier access to these disordered regions. And IDRs are typically encoded by segments involved in alternative splicing. So there's actually, these work together, okay? IDPs or IDRs, post-translational modifications, and alternative splicing work together and function synergistically to regulate cell function. So there's a correlation among these different things, the phenomena. So examples of IDRs in proteins or IDPs themselves include the disordered tails of histone H1 proteins, which collect combination of post-translational modifications, for example, methylation, acetylation, which in turn constitutes what's known as a histone code that helps regulate gene expression. Okay, so we talked about, you know, histone proteins and their tails being modified by post-translational modifications. What we're learning now is that they're disordered, and that they're disordered as well. That aids in their post-translational modification. The disordered KID or kinase inducible domain of Krebs binds and adopts a helical conformation on the KIX domain of CBP, which is Krebs binding protein, triggering transcriptional activation of downstream response genes. Okay. Third, the tumor suppressor protein P53 has a disordered C-terminal domain that can fold into various different shapes as it interacts with different partners, displaying quote unquote conformational promiscuity as it controls cell division. Let's take a look at that in particular with P53. So tumor suppressor P53 shown here, its structure. It has a disordered amino terminus and a disordered carboxyl terminus. And that's shown in this, what's called a PONDR, P-O-N-D-R, prediction of natural disordered regions diagram or graph, okay, for the protein. So you can, and when a PONDR score is 1, that means there's a 100% chance that this particular amino acid is in a disordered region. So you can see here that there's, in general, a disordered region. It's sort of bifurcated by this ordered segment at the amino terminus. And then a central region, which is ordered, and then a disordered C-terminal region. And so you can see the PONDR scores are above 0.5 at the amino and C-terminus here. Again, also sort of separated by this ordered region in the middle of the C-terminus, as for the amino terminus. And you can see that at the very tip of the C-terminal region, the disorder region, the structure, the amino acid sequence can fold into different shapes, shown by these different colors, as the C-terminal tip interacts with various protein partners. So when P53 interacts with cyclin A, which helps control cell cycle by interacting with cyclin-dependent kinases, you can see that it folds into this orange shape, okay? But when that C-terminal tip interacts with sirtuin, which is a deacetylase protein, it forms a different shape. When it interacts with a CBP bromodomain, it forms yet a third. And finally, when it interacts with this S100B beta-beta protein, it forms yet a different structure. So you can see that the intrinsically disordered C-terminus of P53 then can interact with different partners and sort of help coordinate different pathways, signaling pathways. That's what they do. That's what they can do, is help to bind different partners and coordinate and regulate different signaling pathways. Let me go backwards, right? So they can act as hubs in protein interaction networks, integrating multiple signaling pathways. That's shown here, P53. So now let me turn my attention to something called proteostasis, which involves the continual maintenance of an active set of cellular proteins present under a given set of conditions. So it's the maintenance of proteins, their folding, their degradation in the cell. So here's on the left is figure 423 from your textbook, showing cellular proteostasis. So on the ribosome, we have, production of a nascent polypeptide, which can begin to fold. However, it might need help there, where the folding intermediate, folds into the native protein. Chaperones are often needed there. And this figure generally shows where chaperones are needed, okay? Now, this folding intermediate could perhaps not succeed in folding and aggregate. Well, chaperones can help with the disaggregation of amorphous aggregates. Okay. The folding intermediate can form a misfolded protein. Chaperones can help remodel those into the proper folding intermediate to fold properly. The native protein can unfold, and chaperones can help with that. And the misfolded protein can then aggregate, right, to form oligomers and amyloid fibrils, which we're going to talk about in just a minute. Misfolded proteins also can be subject to degradation in the cell on the proteasome, the ubiquitin proteasome system, such that the protein is tagged for destruction and is destroyed. When aggregates of misfolded proteins accumulate, a process known as autophagy, which is basically a cellular housekeeping function to destroy misfolded proteins and damaged or diseased organelles. Autophagy will degrade these aggregates to form peptide fragments. So this whole picture together with the folding, unfolding, refolding, and aggregation and destruction is known as cellular proteostasis. So chaperones, right, contribute to the folding of proteins that are partially or transiently unfolded. And we're going to talk about chaperones in just a minute. Right there. But first, I want to talk about the folding process. Okay, so proteins fold rapidly in general in a cooperative process in a stepwise manner. Small regions of secondary structure assemble first, and these gradually assemble into larger structures. And that's shown in this figure 429 from your textbook, where you have these small regions here of peptide sequence folding locally. Okay. So this one here between residues 8 and 15, it is folding into a hairpin, a pair of beta strands connected by a beta turn. This middle segment is folding into an alpha helix. And this third segment here locally is folding into another hairpin. And these two hairpins, so this helix comes together with this second hairpin structure to form a localized sort of motif, which combines with the other hairpin to form this structure, which is somewhat larger. So this is kind of the process that takes place as proteins fold in cells. It's a hierarchical process. So first, these local secondary structures form, and ionic interactions between charged groups that are close together guide these early steps. Okay. Next, elements of secondary structure that are farther apart come together, and the hydrophobic effect is important here and throughout the entire process. It provides entropic stabilization. It lowers the it lowers the entropy. Proteins with more complex structures fold slower. That makes intuitive sense. If it has a more complex structure, it's going to take a while for the protein to find that structure, and it's going to fold more slowly, and actually is more subject then to not folding and forming aggregates. So chaperones may be particularly important there. Domains of the amino terminus fold first as soon as they come off the ribosome. So they're the first to fold. And in general, thermodynamic stability is not evenly distributed across the sequence within a polypeptide chain. Not evenly distributed, and you may have intrinsically disordered regions present. So that's how proteins fold. But many proteins, again, need help to fold properly by chaperones. Chaperones interact with unfolded or partially folded polypeptide chains, preventing their premature aggregation, thereby helping in the folding process. An example of a chaperone, a very important one, is heat shock protein 70. And how it works is shown here in this diagram on the left. So we have a protein forming a folding intermediate, and then the native structure. If something goes wrong, the folding intermediate can not fold, but rather aggregate and form these aggregates. And here's showing an amorphous aggregate. Okay. Heat shock protein can help protein to fold. So either the unfolded or the folding intermediate can bind to heat shock protein 70. Now, what it binds to is the ATP bound form of heat shock protein 70. Okay, after it does so, then another heat shock protein known as heat shock protein 40 will bind, okay, and trigger ATP hydrolysis. So ATP is hydrolyzed to ADP and inorganic phosphate, and then the release of ADP and phosphate. I'm sorry. So once hydrolysis occurs, this will trigger the formation of what's known as the closed form, closed heat shock protein 70 ADP form, which has a high affinity now for the unfolded or partially folded protein. Okay, that so it latches onto it and keeps it there, and then helps it to not aggregate, basically. And it then releases the protein. So what happens is that a protein known as nuclear exchange factor, when it binds to heat shock protein 70, catalyzes the exchange of ATP for ADP, forming this open structure and causing the release of the protein that was unfolded or partially folded, allowing it now to hopefully fold into the native structure. Only a fraction of them do, actually, and some of them rebind to heat shock protein 70. Others then go into chaperonins like heat shock protein 60. Again, which heat shock protein 60, 60 refers to the size of the subunit, 60 kilodaltons, that assembles into these large complexes, barrel-shaped complexes that help protein folding. Now, so this is a chaperone. Okay, these are chaperones that I've been talking about. But some proteins also require what are known as protein disulfide isomerases, or PDIs, or peptidyl prolyl cis trans isomerases, or PPIs. The disulfide isomerases help to break and refold disulfide bonds to aid in the formation of a proper set of disulfide bonds. You can think of it as sort of a biochemical equivalent of beta-mercaptoethanol in some ways. And the peptidyl prolyl cis trans isomerases are involved in converting cis to trans, converting peptide bonds from the cis to a trans configuration. You know, prolyl residues, remember, tend to exist. The X-pro bond exists in a cis configuration, and that can actually disrupt the proper folding of a protein. So sometimes that is the impediment. That's the roadblock. And when you remove that cis bond and convert it to trans, that then allows the protein to go ahead and fold into its native conformation. So both of these two proteins are also needed. These enzymes are needed in some cases. So now let's talk a little about amyloid fibers and something called amyloidoses. Amyloid fibers are associated with amyloidoses, which include the neurodegenerative conditions, Alzheimer, Parkinson, and Huntington diseases, and the neurodegenerative prion diseases, bovine spongiform encephalopathy, BSE, also known as mad cow disease, and Creutzfeldt-Jakob disease, among others. By the way, I thought I corrected this, but Jakob has a K, not a C. I apologize for the misspelling. Now, Alzheimer's is a prion-like disease. Some people say it's actually a prion disease, and we'll talk about that very soon. But in amyloidoses, what happens is that secreted proteins, instead of being properly folded, are misfolded. And these are converted into insoluble extracellular structures known as amyloid fibrils and fibers. The amyloid fibers are actually highly ordered, okay, but they're the incorrect structure. They're unbranched and have a high degree of beta sheet conformation, as shown here on the left. And you can look at that figure 429 from your textbook. This shows the native structure, okay, the denatured form of a protein, and it misfolded or partially unfolded form with the beta. There's these beta strands here in the middle. And so you form this core of hydrophobic residues that associate and form a beta sheet early in the process. So it's a two-stranded beta sheet, a beta hairpin. These can then come together. These cores of hydrophobic residues can associate. They hijack the normal folding process. And the beta sheets that form then associate further to form an amyloid fibril core structure shown here, amyloid fibril core structure, that they then further associate to form protofilaments. So that's what's shown here in figure A, the formation of the amyloid fibril core structure and then protofilaments. Amyloid fibers form slowly, and the onset of symptoms is usually very slow. It takes decades. So for these amyloidoses, right, it's a very slow onset process usually. Alzheimer's disease involves formation of amyloid plaques containing amyloid beta, also known as A beta, peptide, and or the tau protein. So it forms these amyloid plaques of amyloid beta peptide or tau. Now, these may or may not be what causes disease. And we can see here the amyloid beta peptide of Alzheimer's with these two phenylalanine residues that help to mediate the formation of amyloid fibrils. The association of these can help trigger and cause a nucleation of and formation of the amyloid fibrils. Parkinson's disease involves formation of what's called alpha-synuclein aggregates. These alpha-synuclein proteins aggregate into filamentous bodies known as Lewy bodies. And in Huntington disease, there are aggregates of a protein known as huntingtin that accumulate. I'd like to now turn to prion diseases proper and talk about a medical mystery. Toward the end of his life in 1978, the esteemed Russian choreographer, George Balanchine, began to lose his balance while dancing. Later, he began to lose his eyesight, his hearing, and his sense of equilibrium. By 1982, he was incapacitated, and a year later, he had died at age 79. What happened to Balanchine? Well, only after his death, when an autopsy was performed, was it learned that he had developed Creutzfeldt-Jakob disease, a neurodegenerative disorder linked to mad cow disease. And as we will see, CJD is a disease characterized by the buildup of misfolded proteins called prion proteins in the brain. And you can get more information from this NIH website here. So let's think more about prions, okay? They were discovered by Stanley Prusiner, who's at UCSF in California, who won the Nobel Prize in Physiology or Medicine in 1997 for his discovery. There's a Scientific American paper that was published that I'm going to put in your course materials. Prion stands for proteinaceous infectious agent. Prusiner thought that prion sounded better than proine, so that's why this is a little bit transposed, the order of the letters. Now, this idea, right, was originally a heretical idea. The idea that a protein is causing an infection and is infectious, is the infectious agent, was a heretical idea because we've always thought of infectious agents, right, as being either viruses or bacteria or maybe protozoa, but not proteins. But they were found to be the causative agent of certain fatal neurodegenerative diseases, like Creutzfeldt-Jakob and others that we'll look at. We can think of two definitions of what a prion is, a narrow and a broad. Narrowly, prions are proteinaceous infectious particles that lack nucleic acid. More broadly, they are elements that impart and propagate conformational variability. So we can then begin to look for prions more broadly. And identify certain diseases as actually being prion diseases. For more information, you can look in box 4-4 of your textbook titled Death by Misfolding the Prion Diseases on page 134 to 135 of your textbook. On the right here, we can see the histopathology of variant Creutzfeldt-Jakob disease, which was linked again to mad cow disease in Great Britain. And we see two different panels. Panel A shows a tissue slice from a prefrontal cortex stained with the PASS method. And you can see aggregates of plaques, this darkened material surrounded by the lighter spongiform degeneration. Okay, so there are plaques there, amyloid plaques. And then in B, you can see that these plaques stain positively for prion protein. To develop a stain using an antibody for the prion protein, lo and behold, there they are in those plaques. Now, one question is it the plaques that's causing disease or not? So let's look at different prion diseases, okay? Let's look at diseases in humans and in other animals, and then the mechanism of pathogenesis. So Kuru was discovered in the 4 people of Papua New Guinea who would carry out ritualistic cannibalism. They no longer do, okay? They've been convinced not to, which is they would actually eat the brains of their recently deceased ancestors, a very risky thing to do. They no longer do that. Iatrogenic Creutzfeldt-Jakob disease. Iatrogenic means hospital acquired. This is a disease caused by prion-contaminated human growth hormone or tissue grafts or contaminated surgical instruments. Simply autoclaving instruments is not good enough to get rid of prion proteins because they're recalcitrant to or resistant to being destroyed by heat. Variant Creutzfeldt-Jakob disease, possibly caused by bovine prions. That's why there was such a scare with the with mad cow disease that arose in the late 1980s in Great Britain, where millions of cows were slaughtered. And that's actually discussed in that Scientific American article. Gershman-Straussler-Scheinker disease. Gershman-Straussler-Scheinker is a germline, it's caused by a germline mutation in the prion protein gene. So it's inherited. Familiar CJD, also caused by mutations in the endogenous prion protein gene. Okay. There's an endogenous protein in the body called cellular prion protein that adopts an aberrant configuration or conformation. And that's what causes the disease. We'll discuss that in just a minute. Fatal familial insomnia, also caused by germline mutations in the endogenous prion protein and inherited. Sporadic CJD, not inherited, caused by somatic mutation or spontaneous conversion somehow of the cellular to the infectious form of the prion protein known as scrapie. Because scrapie is a sheep disease, prion disease. That's how the infectious form was named. It's the scrapie form. Here's scrapie. It's in sheep. It's caused by infection of genetically susceptible sheep by the prion protein. Bovine spongiform encephalopathy, mad cow disease, caused by infection with prion-contaminated meat or bone meal. So what was happening in the 1980s and prior is that cows were being fed in their feed bone meal from slaughtered cows, and those were contaminated. So that practice was discontinued. Chronic wasting disease found in deer and elk, which was unknown before 1998, but now is known to exist in those wild animals. So the prion protein can exist in 2 different structural states. A benign, non-infectious form, which is mostly alpha helical, known as cellular prion protein, shown here. You can see the alpha helices shown in red, okay? And then the scrapie form, which is infectious. It contains significant beta pleated sheet. We can see here what's going on. Now we've got this beta sheet. Now, this is the actual experimentally determined structure of the cellular form or domain of it. And this is predicted based on a computer-generated model of what will happen when it changes conformation. Okay, so the cellular is converted to the scrapie form, and this involves conversion of 2 alpha helices into a 4-stranded beta sheet. So we see the 2 pink alpha helices largely disappearing and now forming this 4-stranded beta sheet. in the infectious misfolded form. What happens is that the scrapie form propagates itself by contacting normal PRPC proteins, causing a structural transition. So physical contact but from the infectious to the normal causes it to switch, undergo a conversion. And that's how slowly over time, the infectious form builds up, accumulates. And this structural transition can be mimicked or caused to occur in a test tube. So the endogenous cellular protein is expressed mainly in the neurons of the brain and spinal cord. It's actually attached to the outer surface of the cells, of these neuronal cells, and the cell membrane via a C-terminal GPI anchor. I mentioned that, earlier when I talked about attachment of proteins, or when I do talk about it, a GPI anchor. So this endogenous protein is attached to the outer cell surface via a GPI anchor. And the protein is thought to play a role in combating oxidative stress. And in 2012, a study showed that PRPC, the endogenous protein, might play a role in the processing of microRNAs in the cell, but it's still largely not known exactly what the endogenous protein is doing. This is the structural transition that occurs and how the misfolded protein then aggregates to form larger aggregates. So on the left is the cellular form. Again, you see the helices, and then what you see after that is now formation of the misfolded form, where a couple of helices, one helix, helix A, right, and part of helix B have undergone a structural transition to form this beta pleated sheet structure. And then that region mediates the assembly or the aggregation into larger aggregates. Now these last two were predicted by modeling, computer modeling. We don't have experimentally determined structures of these of the misfolded protein or of the aggregate. And this is how prions are propagated. So you can have the spontaneous generation of the infectious form of the prion, right? Or a mutation that facilitates conversion to the infectious form, or you can have inoculation by ingesting contaminated beef, for example. Formation of an infectious molecule. Endogenous prion undergoes a transition to form the infectious form. Well, when that contacts endogenous correctly folded proteins, it can trigger conversion now to the infectious form and lead to the accumulation of the infectious form, the misfolded protein. That happens over time, so that over decades you have an accumulation of the infectious form of the prion inside the brain of the affected individual. So that's how prions work. And lately, the question has arisen: is Alzheimer's disease a prion disease? Is Alzheimer's a prion disease? And I want to sort of argue that it might be by highlighting this paper that was published a couple of years ago by Stanley Prusner and his group at UCSF. In their study, they show that it's prion activity, namely amyloid beta and tau protein as prions, not the accumulation of A beta and tau plaques and tau tangles, that correlate with disease in Alzheimer's patients. Okay, so it's these prion proteins, the prion activity, not the formation of plaques and tangles, that correlates with disease. What happened was they developed a new method that would allow rapid quantification of the A beta and tau prion proteins in brain tissue from autopsied patients who had died from Alzheimer's or other forms of neurodegeneration. So they developed these new methods that could look for these prion proteins and quantitate the amount. It was an antibody-based method. It was an immunostaining type of method. And what they found was that there was an especially strong inverse correlation between age of death from Alzheimer's and quantity of tau prion in particular. And they looked at a particular phosphorylate form of tau, which is known to have prion activity. Okay, let me say that again. Especially strong inverse correlation between age of death from Alzheimer's and quantity of tau prion protein present. The quantity of active tau prions in a patient who died at age 40 was about 30 times that of one who died at age 90. Now, people at age 40 shouldn't be dying of Alzheimer's, right? And if they are, then that means that they have a particularly bad form of it. And what this is showing is that in the form of Alzheimer's that was particularly bad and which had a very early onset, there was a high level of this tau prion protein. So, and that's shown in this graph here, where we have, different patients who died at different ages, right? And then the level of phosphorylated tau normalized to insoluble tau in these Alzheimer's patients' brains, right? And again, the level of the active prion-like form of tau is 30 times higher in the 40-year-old patient. So we see here, look, see that there it is for the 90-year-old, right, at about 0.01. And here it is for the patient who died at age 40, 0.3, which is about 30-fold times more, right? So which seems to indicate that it's this active prion form that is particularly the cause of disease and death. But some questions remain. Why does Alzheimer's progress at different rates in different people? Are higher prion levels in brain samples of younger patients linked to early onset and more rapid progression? Probably. And third, do older patients have less infective prion or a greater ability to dispose of misfolded proteins? That's unresolved. I should note that many scientists are reluctant to accept the idea that A beta and tau are self-propagating prions. They use the term prion-like. However, reports of rare cases of patients developing Alzheimer's after receiving growth hormone from human brain tissue or brain tissue transplants argues that A beta and tau are in fact prion proteins. But I should emphasize that Alzheimer's disease is not contagious. So it's good to keep that in mind. It's not like Alzheimer's is a contagious disease. Okay, folks, that's it. These are the objectives. I look forward to seeing you very soon in class. Look forward to seeing you in class soon for the flipped classroom. And that's it for this pre-recorded lecture. See you soon.